We're going to talk about the blood now. Here we are. Blood circulation. So blood is a fluid, right? And it is circulated through the body because it is the source of all of the nutrients that your cells need to continue to live. The power behind this circulation is the heart, of course. Everyone knows that. The cardiovascular system includes the heart and the vessels. So that's what the cardiovascular system means. It has cardio, that's heart, and vascular is the vessels. The vascular system includes only the vessels, and the circulatory system includes the cardiovascular system and the blood that it contains. So that's just three different definitions. The lymphatic system, which can also be included in this, is a parallel vessel system which mirrors the veins and returns excess extracellular fluid to the circulatory system. So it goes from a capillary bed, the lymph is picked up in lymphatic vessels, that is the same as plasma, and it's the same as extracellular fluid. The functions of blood, it carries respiratory gases such as oxygen and waste gas, which is carbon dioxide. It carries nutrients, it carries hormones, and other proteins. It also carries the immune system, and it helps the body regulate its temperature. So the blood volume in males is about 5 to 6 liters. A liter is about a quart, approximately 1 quart. 1 liter. That's approximate. And females 4 to 5. So the contents of blood are roughly, we're going to see in the next slide, if we take a vial of blood from you, and we spin it in a device that spins it around, right? We're going to end up with a layer of cells. This is going to be red blood cells. Try to get this right. It's going to have a layer of white cells here, and it's going to have plasma up here. Okay, the centrifuge will spin out. The cells are heavier than the plasma, so those will go to the bottom. And then the white blood cells will be this buffy coat here. This is called buffy coat. Okay, so we have the blood cells. Whoa, the blood cells. That's these. These are the formed elements. The plasma, which is the fluid portion, right here. And the serum is the plasma minus fibrinogen. And fibrinogen is a substance used in blood clotting. Okay, as a definition, hematocrit is the percentage of red blood cells compared to how much blood there is. So if we have 10 cc's of blood and we divide that by the volume of red blood cells, that'll equal hematocrit. So in males it's 47% and in females it's 42%. And this is obviously a rough estimate, but that's what they're looking for on the test. You need enough red blood cells to carry the respiratory gases, otherwise your cells will not be perfused properly and you'll get sick. So red blood cells increase the carrying capacity of blood to carry oxygen. Plasma also carries oxygen and carbon dioxide, but not in a high enough concentration. So they think sometime in our ancestral history, red blood cells came from a different lineage and were incorporated into certain mammals, all mammals, specifically to carry oxygen and carbon dioxide. Just an interesting fun fact. So here's our test tube. You know how to draw blood, or how it's drawn anyway. We centrifuge that out. And then here we have the red blood cells here. This is RBC is red blood cells. Excuse me. These are the white blood cells here. Leukocytes means white blood cells, and white blood cells are WBCs. Right? The plasma is the liquid portion. So approximately 45% will be red blood cells, 1% will be buffy coat, and 55% will be plasma. So plasma is straw colored. It's a little bit sticky. You know, blood is kind of sticky, right? It's about 90% water, of course. It contains ions. These are charged particles, such as sodium and chloride. It contains nutrients, sugars, amino acids, lipids, wastes, and proteins. You have the three organic compounds that cells need to grow and maintain: proteins, sugars like glucose, amino acids, which are smaller proteins, and lipids. So there are three main proteins: albumin, globulins, and there are a series of these, and these are part of the immune system, and fibrinogen, which is part of the blood clotting process. The blood cells, the red blood cells, are called erythrocytes. Ereth means red. Cyte means cell. Red cells. Leuko means white. Cyte is cell. And platelets, which are a fragment of a megakaryocyte. Your blood makes these megakaryocytes. These will break up, and then they're called platelets. So we stain blood cells. We stain them, the white cells, to see which type they are. So different white blood cells will accept different types of dye. So we have an acidic dye, which is red, and a basic dye, which is blue. So they look kind of blue and purple. The platelets look like these little fragments here. They don't look like cells, right? These are all red blood cells here. And this is a typical formation. You see them in rows like this, all red blood cells. Okay? Then we have the white blood cells, which are going to stain differently. They're the neutrophils, the lymphocytes, monocytes, eosinophils. There are more than these, but this is just to give you an idea of how they stain. Back to red blood cells. These are the oxygen transporting cells. Red blood cells are not actually alive. Red blood cells are dead. Red blood cells, cells are dead. They're basically rafts, and they're very, very small, 7.5 micrometers in diameter. The diameter of a capillary is 8 to 10 millimeters. I don't think that's true. I think capillary is smaller than that. I'll tell you, this is what is more understandable to me. A red blood cell is 1/3,000th of an inch long, or wide. And that is the exact diameter of a capillary. So I think this is micrometers. Yes. I don't know why that's wrong. Okay, most numerous of the formed elements was the red blood cell. Females have 4 to 5 million cells per cubic millimeter. Cubic millimeter is about that big. Males a little bit higher. Red blood cells have no organelles and no nuclei. They are simply a raft, a dead raft. Okay? Hemoglobin is the oxygen and carbon dioxide carrying molecule. Okay? It's a protein, and it has angles like this, and it has heme molecules embedded in it. Okay? And this attracts these gases. The red blood cell is biconcaved in shape. Looks sort of like a lifesaver without an empty middle. This increases the surface area, and this is important because it will hold more oxygen and diffuse more oxygen out if it has more surface area. They live about 120 days, and they are manufactured in the red bone marrow of long bones. This is what it looks like. As you can see, it's biconcave. That means one concave surface here, one concave surface here. And that increases the surface area significantly, and it can carry more oxygen that way. So moving on to white blood cells, we have different types. The white blood cells are neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So there are 5 types. Okay? Each has a very specific function in the immune system. There are about this many per cubic millimeter. As you saw in the previous slide, there are 5 million red blood cells and 4,800 white blood cells. So significantly less. The white blood cells protect the body from infectious microorganisms. They function outside the bloodstream, mostly in loose connective tissue. Now, diapedesis is a word used to describe how a white blood cell will leave a capillary. It actually crawls along the surface of the endothelium. It has these little proteins sticking out, and it rolls along the surface. And then if it almost like smells something out here, it will then move between 2 endothelial cells into the extracellular fluid and go after this pathogen. And that process is called diapedesis. Okay, there are 2 types of leukocytes according to which type of stain that they absorb. So we have granulocytes and we have agranulocytes. So when they stain, these have granules in them, and you can see them. So it's just a way to distinguish one group from another group. These are the neutrophils, eosinophils, and basophils, and the agranulocytes are the lymphocytes and the monocytes. Now, never let monkeys eat bananas is an acronym for to remember the names of the white blood cells. Neutrophils, never. Let, lymphocytes, monkeys, monocytes, eat, eosinophils, and B, basophils. You'll just have to memorize lymphocytes and monocytes are agranulocytes, and neutrophils, eosinophils, and basophils are granulocytes. So the formed elements, I don't know why they're called formed elements, but they are the platelets, the white blood cells, and the red blood cells. When you go to the doctor and he orders a differential white blood cell count, he is comparing the number of different white blood cells that you have compared to the others. That is to see, if you have a substantial increase in this type, neutrophils have a specific purpose. Eosinophils have a specific purpose, and they react to different pathogens. So if one of these is elevated, you'll know which type of pathogen is present in your body. So now we're going to go through these. The neutrophils are the most numerous. They phagocytize, which means they eat, engulf or eat, and destroy bacteria. Neutrophils, bacteria. The nucleus has these lobes in it. This is how it's identified on a slide. Okay, these lobes here. The granules right here pick up acidic stain and basic stains. So that's what that looks like. The next one is the eosinophil. This is only 1% of all white blood cells. This reacts to allergies and parasites. And this picks up more red stain and has a large nucleus. Basophils are 0.5%. The nucleus usually has 2 lobes. It's hard to see in this particular cell. The granules secrete histamines. So this is also to do with histamines are for allergies. The function, they also function in inflammation mediation, and they're similar in function to mast cells. We'll talk about mast cells in a while. That's all I have to say about that. Lymphocytes. These compose 20 to 45%. They're most important cells of the immune system. Okay? The nucleus stains dark purple and it almost is as big as the cell itself. This fights infectious organisms and it acts against a specific foreign molecule or antigen. So this is the cell that will begin the cascade which will eventually produce antibodies. Everyone's heard of antibodies. An antibody is an army. Okay, we have 2 classes of lymphocytes. We have T cells and B cells. You've heard of these T cells. These are like snipers. It's like 1 to 1. 1 T cell, 1 foreign cell. So it's it's hand-to-hand combat. Okay. B cells multiply to become plasma cells, which then secrete antibodies. So it's B cell to plasma cell to antibodies. They also save a copy as a memory cell so that when you're re-exposed to the same pathogen, the antibodies can immediately be called upon to kill off the pathogen. An antigen is a foreign protein, cellular or not, unfamiliar to your body, which is identified so that it can be destroyed, usually by antibodies. So antibodies are an army, right? So B cells secrete an army, and T cells are 1 to 1. That's the big difference between them. A monocyte, very few of them, they're the largest ones. The nucleus is kidney-shaped, that's how it's identified. It's phagocytic. So the difference between lymphocytes and monocytes are vast. Monocytes are simply cells which identify a foreign protein and eat it. Lymphocytes begin a full-blown attack by making antibodies, which are an army of attacking cells. Platelets are fragments of megakaryocytes. They function in the blood clotting cascade. They are like soft plates of sticky substance, like glue, and they plug up the hole that's made in the capillary or artery. So this is a great summary of the formed elements. Here's your red blood cells. Here are the white blood cells, and here are the platelets. It gives you the numbers, the descriptions, and the functions. You can look that over. Another definition, hematopoiesis is the process by which blood cells are formed. Some say 100 billion new blood cells are formed each day. Now, if the kidneys, which are responsible for monitoring how many red blood cells are in your blood, if it detects that you have too few, it will notify your brain, and then your brain will send out a hormone called erythropoietin. That's monitored by the kidneys, and the kidneys will report, and the signal will come back, and erythropoietin will be put into the blood, and that'll go to the bone marrow, and the bone marrow will release red blood cells. So bone marrow is the site of hematopoiesis. It's located within all bones. Red bone marrow actively generates new blood cells and contains immature erythrocytes. They remain in the epiphyses, the girdles, and the axial skeleton, or of the axial skeleton rather. The yellow marrow, remember that? It's dormant and it's mostly fat. It's located in the long bones of adults and has a tissue framework for red marrow, and it's made of reticular connective tissue. So all blood cells originate in bone marrow. All originate from one cell type, a blood stem cell, which is a pluripotent. That means it can develop into many different cell types. Pluri means many, potential means may become. We have lymphoid stem cells. These will give rise to lymphocytes, and myelostem cells give rise to all the other blood cells. Erythrocytes start their lives as pro-erythroblast. Remember, blast is the precursor cell. They remain in the reticulocyte stage for 1 to 2 days and make up 1 to 2% of all erythrocytes. Leukocytes form from granulocytes from the myeloblasts. I have a chart of this, hang on. The platelet-forming cells are from megakaryocytes blasts. And here's the chart. So you have it's an undifferentiated blood stem cell, so it's a stem cell, but it can only make blood cells. And that will give rise to two different types of cells, the myeloids and the lymphoids. So the myeloid will make all the cell types except for B and T lymphocytes. They have a different lineage. Okay, then we have this megakaryoblast. It forms a pro-megakaryocyte and then a megakaryocyte. That'll bust up into little pieces called platelets. Then we have the monoblast, pro-monocyte, monocyte, and etc., etc. You can look this over. It's an easy way to see where these cells came from. So disorders of red blood cells, you can have too many, and that would be called polycythemia. You can have too few, which is called anemia. You're familiar with that, I'm sure. You can have sickle cell disease, where the shape of the red blood cell changes into a sickle cell formation, and it can't carry enough oxygen. It's an inherited condition, results from a defective hemoglobin molecule. Erythrocytes distort into a sickle shape. Then there's hemochromatosis. This is inherited, and this is abnormal excess of iron. Disorders of leukocytes, of course, leukemia. This is cancer of white blood cells. And disorders of platelets are called thrombocytopenia. That's a low concentration of platelets, and that means that you will have a bleeding disorder. You will, every time you get a little cut, you'll bleed. And you don't want to bleed because you'll lose all of your blood, right? So, the first blood cells develop with the earliest blood vessels. Mesenchyme cells cluster into blood islands. Late in the second month of gestation, the liver and spleen take over the blood formation, and then that will migrate to bone marrow at about 7 months. That's all you need to know about that.